Yes, sir, you're live now.
All right. Good morning, everybody. Welcome to all the students who are taking MOOC courses, especially the Proteomics and Proteogenomics course. Today, we are going to talk about a very exciting technology, mass spectrometry, which has become the major working horse for the proteomic applications. As you've been already studying in your course, that how mass spec has revolutionized our ability to look at the complex proteome, its protein identification and quantification to a scale which was not possible earlier. Earlier, when we started our conventional 2D gel-based approaches, I hope you recall those techniques, where most of our the major hits were coming from the gels with all these spots which we were seeing them on the 2D gels, right? But then moving forward, idea was, can we take the samples, whether it is coming from the tissue, body fluids, or any kind of biological sample, and rather than separating them on the gels, can we directly separate them with the different properties using mass spectrometry? So use different type of chromatographic principles followed by mass spec analysis. So that became shotgun proteomics approach. This entire trajectory of changing the field of proteomics, uh, reaching till the to the field of shotgun proteomics was all happening in the 1998 or 2000 kind of years. That's the time when 2D gel and the field of DIGE was getting developed. And then people realized how challenging it is to get the good quantification data from the gels. Because of the reproducibility issue that we have been discussing in the course, that's not very easy to do the high throughput proteomics on the gel. Then people realize, can we utilize the strong cation exchange and reverse phase chromatography, the two dimension of separation of the protein, and directly separate those chromatographic peaks and analyze them on the mass spec and use that information without gel. That was known as the first paper on the MUTPIT, multi-dimensional protein identification technology. And then field progress forward, because then idea was, we still want to quantify the signal. We want to still look at comparison of our control versus disease, and how to do all that if we are not running on the gel. Can we look at different condition, different quantification also on the directly mass spec based approach? But the current state of the art is that you can take any samples, including people are not even targeting single cell, to think about how applications can be done on the single cell level, especially when you think about the cancer tumor heterogeneity and many other biological applications where every cell itself is having a lot of its own property. And when you pool the different type of cells together, it may not give you meaningful data. So if one could take those any biological sample, directly extract the protein out of it, and those lysates are being digested to make the peptides. Especially you use the enzyme like trypsin, chymotrypsin, lysine, many type of enzymes you can use. And then followed by, you separate them on the liquid chromatography, and then you want to do the mass spectrometry based analysis. This is quite important point which is now shown here in a recent review article by Matthias Munn team. There are two different approaches of now how you can uh, proceed for mass spec based analysis. Either you can use the DDA, data dependent acquisition, or you can use the DIA, data independent acquisition. Uh, so these two approaches conceptually have a lot of importance. For example, in DDA, you're looking at the most intense precursor, like what you're seeing in the red, the green, the blue. It's only picking up, the mass spec is only picking up these most intense precursors and trying to fragment those in a specific, millisecond time, and then goes to the next round and again picks up the most intense precursors and then try to further fragment those for MSMS data generation. So that is the DDA-based approach which has been conventionally used, whether we talk about our label-free quantification or we talk about TMT or iTrack. All of them works on this principle of looking at the top N precursors which are going to get fragmented. Then the newer approaches came, which is known as the DIA, data independent acquisition, where idea is, since we have peptides coming and eluting in different M by Z, starting let's say from 300 to 1300 M by Z, where most of our peptides will be seen. If we can take the small windows of 300 to 400, 4 to 500, and likewise we go to let's say 1300, and what you see now, those colors, like you are looking at every precursor in that M by Z, not only picking up the most intense ones, right? So you are now looking at the entire mass and charge window and trying to utilize that information irrespective of looking at only the top end. So this way, your coverage is becoming much broader. Of course, there's a lot of intricacies involved in doing DIA, whether need to do the library-free approach or library-based approach. How you're going to analyze your data becomes much more challenging in the DIA. Although there are newer approaches which are being utilized, software commercial like Spectronaut is there, but there are more free softwares like we have the DIAN, DIA-N, DIAN software. We have the FRAC pipe. So some of these softwares have really started making the DIA analysis quite easy, and therefore your coverage can really jump up quite well. And the bias of the missing value, which we see more in the DDA data, where a lot of peptides are not getting sufficiently captured, those are known as missing values. That issue has been quite well resolved in DIA. So these are the kind of, as I said, this is theoretical part. This can be quite important, and it has a lot of detail. Additionally, one could now look at validating your targets and do the targeted and NDIA, different type of analysis. Targeted proteomics itself is having a lot of interesting thing to talk, especially the two workflows of MRM, multiple reaction monitoring, and PRM, parallel reaction monitoring. And all of that we can try to look at a specific peptide, and we want to validate those peptides. So that we'll talk as we go along. Finally, then in this data, what comes forward is you get some sort of, the heat maps and the various ways of principal component analysis to find out differentiating your control and treatment, how well they can be differentiated, as you can see in this type of plots, where you in the volcano plot, you can see like, the proteins which are passing the threshold, which are extremely up or down, and significantly they're passing the threshold, those will be considered forward for your network analysis and the interaction analysis likewise. Yeah. So at each level, there are a lot of automations are happening, a lot of advancements are happening in the this new field of mass spectrometry-based proteomics, where a lot of robotics is being utilized to increase our throughput for the sample prep and looking at different ways of our speeding up the process and bring the more reproducibility. Different type of multiplexing possibilities are being also utilized, especially different type of chemical labeling approaches. Liquid chromatography is enriched with various type of new columns and different type of preform gradients. Especially EvoStep is one of the currently the big leader in the industry with advanced lot of our chromatographic experiences. And then there are different type of mass spectrometry instruments which are being used, which I will talk as we go along. So we don't want to make this again as a theory lecture and session right now. Idea was to give you uh, some update about the field and also show you live from the lab how different type of experiments can be performed. So TAs are going to take lead and they're going to show you different steps involved. So first we are going to talk about you have extracted the protein, how you're going to prepare the peptides. So if you have to do the gel, then you are doing the in-gel digestion. But in this case, where you are going to avoid gels, so directly we are going to take the protein lysates in solution form and do the in-solution digestion. And once we have achieved that, then we would like to know how much quantity we have achieved from our peptide sample. The peptide quantification will perform. So both of these will be our hands-on session now, which we are going to do. Followed by, we'll take these samples and do the mass spec analysis as we go along on different instruments. So I request now TAs to share the screen and proceed for the hands-on session on the peptide preparation in solution dilution. Amit, you are not audible. You have to unmute.
Hello, am I audible? Hello?
Start
Yeah,
again. Yes, now you're
yeah.
audible.
So good morning all. So welcome to the MOOC live session. I am a TA for the Introduction to Proteomics course. So today we are going to basically know about that how we can digest the proteins into peptides. Digest the proteins into peptides, and that we can analyze for the mass spectrometry analysis. Now, Now here you can see the lysates that we prepared in the last day, and now these are the proteins that are available in the lysates. Now the very first thing is that whenever you are going to start the digestion, you need to understand that how much protein that you need to first digest. So coming to the you can see Excel sheet here. So in the Excel sheet, you can see with the help of Bradford quantification, we have already measured that what is the concentration of the proteins that is present in the lysate. So today we are going to digest the protein sample that is present in this sample, UNDIL3. Now you can see this is the concentration. The 6.72 microgram per microliter is the protein concentration. And for digestion, we need almost 30 microgram of protein samples. Now, before starting the protein digestion, you need few important ingredients that will help in your digestion. The very first important thing that you need is the buffer B. So this is the buffer B. Actually, you need to make up the volume for the buffer B. Now you need to use the TCP. So TCP is a important gradient that will basically break the disulfide bonds. After TCP, you have the IA, the iodoacidamide. So this is the iodoacidamide you can see, and this is very light sensitive. You need to make them fresh whenever you are going to start the digestion. Now, what is the role of IA? So IA basically helps in the alkylation. It will prevent the reformation of the disulfide bonds. Now, and one of the most important ingredients is the trypsin. So trypsin, we all know that is the protease. It will basically break after the lysine and arginine. And question is why you use trypsin? There are also many proteases are there, right? We can break the long chain of proteins. So basically, trypsin will break in the arginine and lysine residue. One lysine is one of the most prominent or predominant amino acid that is widely present in all the protein molecules in the amino acid stretch. Now, the very first thing is that you can see that total 30 microgram of protein we need. So we will take the sample here. Now, you can see here the total 2.97 of microliter. That total 30 microgram. The 30 microgram protein is there. Now we will take only 3 microliter, 2.9 microliter of protein from the lysate. Yeah. Here we're putting the samples. Now we need to make the volume with the help of buffer B. Now buffer B, we need to make the volume up to 20 microliter. So 20 minus 3, that is 17 microliter of buffer B we need to put. We will keep the buffer B here. I will mix it well. And now you have the mixture of the buffer B and the lysate mixture. Now you need to add the TCP, which is basically useful for breaking the disulfide bond. So we take 1.2 microliter of TCP. And after mixing every component here, you need to show that you will vortex it well. So basically, for after adding every ingredient, there are some incubation time. Like even like one of the examples is that trypsin, which is caused by which we have to keep it for 37 degrees Celsius for overnight incubation. Now after adding the TCP, we need to keep it for always the 20 minutes of incubation. After we can add the IA. So IA is basically the compound that will prevent any kind of alkylation. So for right now, we will add the IA, and after that we will add the trypsin to start the first round of digestion. So here we are using two different two-step digestion. Here we will add the trypsin for in 2 times, and we will keep the solution in 37 degrees Celsius. Now I'm going to add trypsin. And also after adding the trypsin, we need to also keep the solution, the pH of the solution 8. So for that, we will again need to add buffer B, and we need to check the pH. So actually the in solution digestion is quite a very long process. You need multiple ingredients, and you need to keep the solution in for 37 degrees Celsius incubation for an overnight incubation. Now, whenever your digestion is complete, you need to also acidify the solution with the help of Fa, the formic acid. And after with that, you need to first dissolve the entire solution. So in this entire procedure, we will use a lot of salts out there. So you need to also get rid of the salts. So with the help of these cartridges, you can get rid of the salts from the solvents or the solution. Now, after entire process, you will have the peptides here. You can see the very little bit amount of peptides here, and you need to add the Fa to again resolubilize the peptides. So for desolubilize, so after that, we need to find out that what is the amount of peptide concentration is there. So for that, you need almost two different kind of wavelengths. One is the 205 nanometer, and is the 280 nanometer. So whenever you get the dried peptides, you need to resolubilize the paper with the Fa, and after that, there is a particular micro droplet that I will show you. Yeah. So this is the micro droplet. You can see the peptides we need to put in this micro droplet, and uh, with the help of this instrument, we can easily find out that what is the amount of peptide out there. Now we are going to put the peptide solution inside the micro droplet. So I will be taking 2 microliter of samples. I'm going to keep it here. Simply again, I will be doing in replicate to minimize any kind of variation. Yeah. So again, before starting the experiment, we need to also set the blank here. So blank, we will use the Fa as a blank. I'm keeping the blank here. So you can see the drops at the micro droplet. Yeah. So now I will close it. I will close the lid. Yeah. So your micro droplet and your samples are ready. Now I'm going to share, and I'm going to show you a software which is called the Multiscan Go. So now we are going to keep the micro droplet inside the Multiscan Go. Yeah. So you can see the plate is already out. I am keeping this microdroplet here. Now we are going to, you can see there is a plate layout here. Now we need to first set the plate template here. So this is a microdroplet. You can see thermoscientific microdroplet. I'll choose it. Then I have already put the two blanks here. So I'll keep blank in these two tabs. Then I'm going to understand, I'm going to find out what is the concentration of peptide. Now I'll keep set unknown here. I'm keeping in replicate, and I keep that in B. Okay. So now I have now set the plate layout. Now go to the protocol. Basically, in protocol, you need to set the absorbance. Absorbance, you will see use multiband wavelengths, 205 nanometers, and another wavelength is 280 nanometer. We'll keep precision. We'll go to absorbance. We'll perform blank subtraction, and then we will start the sample acquisition. Yeah. So after that, it will take few minutes, and it will start the peptide. We will get the absorbance of the peptides, and we will try to, and you can easily calculate that what is the amount of peptide concentration that is in our sample. Yeah. So you can see there total 1.494 is the absorbance, and 1.515 for the sample for the another replicate. So basically, you can export this entire information. So you have already this information. You can basically so this is the amount of 1.4 and 1.5 is the absorbance in the 205 nanometer, and 0.057 is the absorbance at 280 nanometer. So basically, you can keep this take these values and paste it in a particular Excel sheet. So basically, for this kind of peptide quantification, we use Scoop's law, where we use 205 nanometer and 280 nanometer and try to find what is the extraction coefficient of the 205 of the sample. Yeah. So now we'll basically drag it down. So there are total, you can see there are total the concentration of peptide is 0.6 microgram per microliter. And now if we talk about orbital fusion, for the in kind of global protease, we at least need 1 microgram of peptide. For a safer purpose, we generally give the machine to 3 microgram of protein peptide samples, and we also make up the volume with the help of FA. So thus, you will come to know that that's how so starting from Bradford quantification, you can see what is the amount of protein is there, and after using the trypsin and following some of the steps, you can also see how much peptide is there, and based on the concentration of the peptides, you can put the samples into the mass spec, and that you can go for any kind of global proteomics acquisition. So yeah, this is for the insol digestion and the peptide quantification for any kind of insolation digestion.
Okay, so now I think the samples are ready. So we can try to take the sample and proceed for the mass spec analysis. So TS will now take the peptide and will go shortly to the one of the instrument which we have, which is high throughput proteomics instrument for doing the deep proteome analysis. Most of the time, your goal is first to look at total how many protein can we take from our that sample, right? So in that way, you are now going to do the deep proteome analysis, and there could be different workflow as we talk DDA, DIA. Even within DDA, one could go with the label-based approach or label-free approach. Label-based approach means you have, let's say, multiple conditions to compare, and you can label them with different type of isobaric labels. For example, tandem mass tag or isobaric tagging for relative and absolute quantification IFRAC. and you can use these reporter ions and label with those type of set of the multiplex isomeric tag of TMT or ITRAC. And then you can pull them together and you can do the high pH fractionation. And then each of those fraction you can run separately. So that's one way of doing the proteomics, quantitative proteomics. Other was the label-free approach, where you're running every sample separately, and then you are observing their entire chromatogram pattern and hoping that retention time is quite well maintained by spiking in some of the IST peptides, international peptides. And then you are going to compare the data and look at their overall intensity, they're looking at the spectral count, and based on that, you can analyze the data. Or the third approach, as I said, it could be DIE, data independent acquisition, which is the new workflow which is being optimized. So all of this can be applied. And now TS are going to show the instrument, Orbitrap Fusion, which is one of the tribrid instrument. And we primarily use for a lot of deep proteomic based analysis and quantitative analysis. So Amit and Ankit, please discuss about the orbital fusion now.
Yes, sir. Thank you, sir. Yeah. So, hello everyone. Good morning, everyone, and welcome to this live session. So, I think Amit has shown, right, like we have taken the samples and have performed the peptide quantification. And now, if we want to do the mass spectrometry analysis, so these are the tubes or vials in which we keep for mass spectrometry that sir was mentioning about the orbital fusion or the tandem mass spectrometry. But before you know that before going into the mass spectrometry or the MSMS, so it is actually LC MSMS that we generally talk about. So LC here is nano LC or nano liquid chromatography. So you know that what peptides is from the complex samples, like from a tissue, cells, we get it contains a complex mixture of proteins, then which we are digesting it into peptides. Now we want to see that what are the peptides that are present and quantify them, right? So this cannot be achieved if we inject all the peptides at one go, because the detector and the mass spectrometer, it will get oversaturated, and that's why what we need a kind of peptide separation, which we do using reverse phase chromatography. And for this instrument, we use nano LC or nano liquid chromatography. So why it is nano? Because I would like to, so we can focus here. So you can see the flow rate. It is in nanoliter per minute. So waste flow rate comes in a nanoliter per minute. So you can understand that it flows in a very in a very small volume quantity over the time, so that the peptides, complex peptide mixtures that we have got, can be easily separated out. Now, this I would like to show that this contains the two solvent systems using two pumps. Here we make the gradient. So one contains acetonitrile, one contains 0.1% FA water. So with that, the gradient of 40%, 50%, 60%, 80%, all these gradients are made so that the peptides can be eluted as per their hydrophobicity. Like in the 2D and the gel molecular weight and pi is used, here we are taking the another peptide characteristic, that is hydrophobicity, to be used. And these two solvent systems, this chromatography is helping to create the gradient to which it gets eluted out. So these samples that we bring, so what we do is that here is an auto sampler. So just, yeah, so here is the auto sampler where we just put it up, okay, and then we have just eject the tray. So now once we eject it, the tray, so now we can give the command, the instrument to take. Before going into the parameters and those, I'll just like to show like what Sarah has mentioned. So this is the mass spectrometer which is connected to this nano LC. The peptides are getting injected using this column. So this is a pre-column followed by a column. So column is nothing but a pack of C18 material that binds the peptide, and then as per the in the stationary phase, and when the mobile phase solvent comes, so the peptide gets eluted out and gets injected into it. Now as so you know that now we talk about LC MSMS, so tandem mass spectrometer. So MSMS means there should be two mass specs because we want to get the single sequence or the amino acid that is there. So now this instrument gives, like, there are three mass analyzers present in this instrument. So one is quadrupole, which we need to segregate the first precursor ion, first kind of window where we are filtering it out. Next, it contains two more mass analyzers are there. One is orbit trap and the other is ion trap. So you can either couple quadrupole with ion trap or you can couple quadrupole with orbit trap. And in between there is a C trap which actually captures the ions and then give then according to the method and the parameter that you give, so it will distribute the ions there. So this gives us an enhanced separation with a superior resolution and a greater select sensitivity. So this instrument gives you a kind of, you can go to a very high 120,000 kind of resolution with a greater speed and accuracy. Yeah. So now I'll just show you something. So yeah, so when you will be doing, so they will be getting this kind of chromatogram. So you can see that if I just try to show you, so it will start from 0 to 120. So 120 means that we are giving a 2 hour long gradient where the peptides are being eluted out. So here the from the we are going from lower hydrophobicity to higher hydrophobicity, and the peptides are eluted out at each phase. So while the peptides are eluted out, it is going to the detector and we are seeing the intensity. So you can see here that the intensity is coming in the range of E9. So the maximum intensity of the precursor that has got is E9. But this is the total ion chromatogram of the MS1. Now, if you want to go and see that a particular, so these are all you are getting are your MS fragments. So for a particular precursor, what are the fragments? So once for any precursor M by Z, that is for a total molecular weight of the peptide followed by the charge and its MS, the fragments. Then the fragments are tried to map with the particular monoisotopic mass of a sequence of a peptide, so that we can get the stretch of the sequence of the complete amino acid which are present in a particular peptide, and then finally bioinformatically peptides are matched with the protein to identify the complete what is there. And these intensity values of the peak are actually led to the quantification and how much proteins are expressed in a particular cell. Yeah, so this is in short about the orbital fusion, like starting from the connected to nano LC. Now we can move on to the next instrument, which is another orbital fusion, but Q exactive. Yeah.
All right, thank you, Ankit. It was a good overview of orbital fusion. So again, mass specs can be utilized for different type of omics-based investigation, not only the proteomics, but also it can be utilized for metabolomics or lipidomics, right? So one can always try to utilize the power of these instruments and use them for multi-omics investigation. And even from the same sample, you can think about whether we can, even try to do this, multiple layers of the sample. One could extract from the protein, one could do the metabolite, one could do lipid, and then try to build the layer-wise information. Of course, people also do genomics on the same sample. So then one could do truly integrated omics, in that way. So while this instrument, what we are going to show you now, which will be another Orbitrap instrument and the hybrid with the, when we can do the things for proteomics as well as for the metabolomics, but we primarily use this instrument along with the UHPLC for doing the metabolomics application. And here we have the quadrupole Orbitrap, that's what is hybrid, unlike the fusion, which is the tribrid instrument, right, which with the ion trap was added over there. Metabolomics workflow is quite straightforward. You can use some organic solvent to extract the layers for doing the metabolite as well as lipids, and followed by acquire the data using this LCM-SMS setup, and then do data analysis using different type of databases. So now over to Amit, and he will show you the Q-executive Orbitrap mass spec and talk about the metabolomics workflow and data acquisition for that. Amit, you can take over.
So the second aspect that we are going to talk about is the Q exactive. Right now we are doing both untargeted and targeted metabolomics and lipidomics too. So as Sir said, the sample processing is quite very straightforward. You will have the chromatogram and you will get the layers for both the metabolomics and lipidomics. Now we can see this entire the mass spectrometry and there is the HPLC, the high pressure liquid chromatography is coupled in this instrument. In that system, you see LCMS, here is the HPLC is there. and the model name is Ultimate 3000. So you can see this HPLC is quite different from the NanoLC. Okay, so there are 1, 2, 3, 4. 4 different compartments are there. The very first compartment is the column compartment. So here for lipidomics we use CSH column, and for metabolomics we use kind of C18 hydroxyl gold bond. Now one of the very important feature, the technical feature of this column compartment is the thermostatic mechanism. So basically it provides the passive and active heating of the columns. So for the good coverage and to also to ensure there is no RT sheet of the samples or the metabolites or lipids that you are going to see, you should not compromise with the temperature. So this kind of component will keep the atmospheric ambient temperature for 40 degrees Celsius, and it makes sure that there is no such fluctuation in the ambient temperature. So basically for this kind of columns functioning, the column compartment keep the temperature for 40 degrees Celsius. The second compartment is the auto sampler here. So in auto sampler is essential because of the reproduction automated sample injection in the QE. So you will see there are three different trays are there: the green tray, the red tray, and the blue tray. And these are the different vials where we need to keep the samples. And inside it, you can see the insert here. So basically, in this kind of metabolomics or lipidomics, use this kind of plastic vials. Now, in one tray, you can keep 40 different samples. Okay. And for entire sample queue, there are 3 trays are there. So there is 14 to 3, 120 samples you can keep for the entire sample queue. And one of the most important technical feature is that it has lot of wash pores out there. It will get rid of the any kind of the contaminants, and it will also get rid of any kind of carryover if that is coming through the sample when the sample is when the sample acquisition is happening. The third compartment from the bottom is the pump compartment. So the pump compartment, let me open this pump compartment. So it will give us the feature of the pulse-free flow. So it means there will be uninterrupted flow of the solvents, and it will also get rid of any kind of back pressure fluctuation. This is another important thing that you can see. It is a knob. It is basically for the purging system. So basically, whenever you try to give the new solvents for any kind of acquisition, whether it's untargeted or it's targeted, you need to get rid of any kind of bubbles in the solvents. Okay, so basically you can just use these kind of knobs and you can start purging the system. So basically, if they have a purging, you will see all the bubbles are getting out of this particular pipe. Now the final rack is the solvent rack. So in case of lipidomics, you use a quaternary solvent system, and in case of metabolomics, you use any kind of binary solvent system. So in case of lipidomics, we use 4 different solvents out there: the solvent A, B, and C, D. So one of the most important feature of this mass spec is that it can give us the information about the protonated and the deprotonated metabolites or even the lipids. So in case of positive mode aggregation, we need to use a solvent A. That is the composition of and the composition of the solvent A is the ACN and the water, and the solvent B, it is the composition of the IPA and the ACN. Similarly, in the case of negative mode, the similar kind of solvent is used, but there is no F. We do not use any kind of FA, but we add only the FA in case of the positive mode aggregation. Now from the HPLC, we have our one of the most important mass spectrometer here, that is the QE. So as sir said, there are 2 different mass analyzers. There are 2 different mass analyzers are there. One is the Orbitrap and one is the Quadrupole. So the very first thing you need to know that you are in the courses you have already learned about what are the hard ionization and soft ionization technique. Within soft ionization, there are techniques like MALDI and ESI. So it uses the API, atmospheric pressure special ionization, which is the ion source, and the mode is the ESI, electrosphere ionization. So in case of MALDI, you will get to know that only single charge ions are generated, but in case of this ESI source, there are multiple charges are getting generated. So with the ESI, there is this entire thing is called Hazy-2 probe. So Hazy-2
Amit, I think
is
just going more details than what time we have. So
okay.
Thanks for this demo. I think we should move on to the next concept, which will be the targeted approaches for looking at the proteins and metabolites. So QE, as you have already heard, can do multiple things, and we feel this is one of the very robust instruments for doing multiomics. But of course, once you have identified your good targets, whether it's the peptides, protein coming from the, let's say, Orbitrap technology, or you have identified some metabolites and lipid using the another instrument. So then now you want to validate those, and then you can come to the triple quad based instruments, one possibility, or you can also use Orbitrap instrument for another possibility. The two different workflow. One is known as the multiple election monitoring, which you can use primarily with the triple quadruple instrument. And you can also do the PRM, parallel election monitoring, which you can do from the Orbitrap technology. So we are going to now show you how to perform the target analysis. Idea is that you already know your which protein and peptides or the metabolites of your interest. You are only going to provide that list to the software, which now instrument is going to screen. Software Skyline is being primarily utilized for this purpose. And then those selected precursors are being fragmented, and you're going to generate their intensity profile and use that, and then try to correlate whether your discovery data and validation data are matching or not. So we are now going to discuss the triple quadrupole based instrument and technology for doing target application. So over to Ankit now.
Yes, sir. Thank you, sir, for the brief overview of the targeted proteomics and the instrument. So just now what I'm going to show you. So I've shown you the future and Amit has shown the QE. So now what we are going to show you, one of the instruments is the triple quadruple base, that is TSQ voltage, and it is again coupled with an HPLC. So this is not coupled with nano LC like the one in the future, but this is what we are. Yeah. So what we are coupled with is like with the HPLC. So why generally what is the main difference between nano LC and HPLC is the flow rate. So HPLC we get the flow rates in ml per minute, whereas I have shown in the nano the chromatography we need in nanoliter per minute. Since we are here just going with a few target peptides and target metabolites or lipids, whatever be that. So that's why we are using in a high flow rate so that we have a less gradient, we use less solvent, and we can get accurate quantification of those particular peptides. So I'll just show you, this is another model of the HPLC which consists of the Vanquish. So here again you will get the chamber of the auto sampler where you can put up some solvents, and this is the pump system, and this is the column compartment. Now with this column compartment, it is attached the triple quadruple. Now if I going to say what is the major difference between triple quadruple mass spectrometer. So one thing is that, so here we are talking about the different mass analyzers. So we need two mass analyzers. Here just focus on the name, that is triple quadruple. But here there is not that there are three quadruples. So don't get mistaken by the name that it contains three quadruple. It contains two quadruple, and in the middle is one collision cell. So it contains one quadruple. So we have already spoken that the quadruple contains the filtering of the precursor ions. Now there is a collision cell in between, and there is a third quadruple. So how we generally give this kind of instrument, because these cannot go like you can collect all the whatever is present in your sample in a global way. Rather, we have to give the precursors that will be filtered out in the first quadruple, as well as the product ions after the collision energy collision cell that we are getting to identify in the third quadruple will also we have to give out. So why this is known as targeted, or it is specific and precise? Because we are giving the quadruple one precursor and its product M by this. So it will capture the exact specific for a peptide and metabolite. So that is the actual, we can say, as a fingerprint for that particular peptide or a molecule and a lipid. So that's why you are very precise. So I'll just show you a quick overview like how it is generally we are done. So this is kind of a, I can show you for a molecule how we measure. So this is done using the Skyline software. So for example, this is one of the compounds. So here you can see that it is the molecular weight, so M by Z of the precursor. Okay, and these are the products. So we had instructed the instrument to capture each of these precursor and its product specific for this particular molecule to identify, because it is quite signature to this particular molecule. So now after the data acquisition, you will get this type of graph where you can see this is the retention time. So at 5.5 minutes, again, this is coupled to a C18 column. So where the hydrophobicity is used using the different solvent system to generate, and at 5.5 minutes, you could see that it has, or at a 5 minutes, sorry, yeah, 5.2 minutes, yeah. So it has eluted out and it got the peptide. So in this particular retention time, so you have to see that whether there's a retention time shift or not. So Sam was also mentioning about different kinds of QC checks using different IRT peptides or different kind of standard metabolites. And this is the intensity which is getting around that, what is the peak observed under the area. So since this particular will give you the areas of all these particular transitions, it will give us the quantification that what is the actual intensity. But it is, as I have shown, it is quite specific because we can measure the precursor peptide, precursor product pairs, and that's why it is unique and known as the targeted, and we can exactly precise whatever the list we have identified in global can be validated here. Yeah.
All right.
So,
Thank you, Ankit. So I think, student participants who are available online, uh, what all different type of theory you have studied already. Now you can see their actual practical aspect of it, and along with the instrument demonstration. Now what we are going to do, we are going to show you a new technology, which is one of the latest in the field, which is the timsTOF technology, which actually does the 4D proteomics analysis. And fortunately, at our startup in collaboration with Bruker, now we have acquired this technology of timsTOF. And I now request our team at Proteomica to share their video and do a live demo over there to talk about the timsTOF technology, which we think is giving us unprecedented scale of the data. What typically, again, in the proteomics, our goal has been to first get as many protein as possible from a given sample. So using different workflow of DDA to DIA, what we have been trying to achieve was around 5,000 protein from a given typical cell line sample. But now where we have reached with this new technology, almost now 8,000 protein, 8,000 plus proteins very easily, and the group analysis even crossing the 10,000 plus protein. So this is a wonderful, incremental change what we could observe now using the 4D omics technology of the timsTOF. So request Dr. Argya now to show the timsTOF instrument and explain about it.
So welcome everyone. So what you are seeing is the India's first TIMS facility here. So just to give a brief background, you have seen the mass specs which can do a three-dimensional separation right in the form of RT as well as M by Z as well as intensity. This is the first mass spec which is timsTOF Pro, which can do a separation on the fourth dimension. That is based on the cross-sectional area. So let's move on. I will explain the later. So this is what you are finding is the timsTOF Pro instrument coupled with the EvoSep Nano LC. This EvoSep Nano LC is one of the cutting edge revolution in the world of Nano LC. Here you don't have to keep different samples here. You just have to put this kind of tips, which are Evo tips. You directly load the samples and the samples will be directly eluted. Rest all the things are like the general things. You can see the pumps. Through the pumps, once the sample is injected, is brought here. From the sample injection, it goes to the column. Like you can see, this is the column here. So this is a 15 centimeter column which is being attached. This is the column toaster. And now, once the column has been done, it is getting injected into the mass spec. So what happens? You have to think like a ship. Like there is a ship which is sailing. The ship which has longer pals can capture more amount of air and it can move faster. Similarly here, the bigger ions which are there, it will move faster. And similarly, the smaller ions will be left behind. You will have a drag force also. At a certain point of time, it falls down and then it goes to the timsTOF Pro the top detector. So you have to understand there is no exclusion of any ions. All the ions which enters into the timsTOF Pro goes into it. That is why its sensitivity and specificity is very high. You can see 800, you can see like 8,000 proteins with only 200 nanograms of protein. So this is the software. What you can see. This is how the samples are being run. Going to the quick elution viewer, so you can find this is one of the DDA. Let me show you. Okay, so this is one of the plasma serum samples. You can see the chromatograms. This is how the chromatograms look like. And when we have gone to the online protein detection, it can yield, like, just imagine 200 nanograms of serum samples or the plasma samples have the capacity to elute around 1208 proteins, like more than 1000 proteins, which is more than the conventional proteins. Similarly, we can see the HeLa also, like this is the HeLa, the cell lysate pattern. From 300 nanograms of the peptides, we can easily get 7816 proteins, like more than 7,000 proteins. So this kind of cutting edge technology is now revolutioning. The most important thing is, like, you can store the samples, you can analyze after 3 6 months. As we have the property of ion mobility and the cross-sectional area, the sample, if it is degrading or something happens, but the areas will be the same. So reproducibility is very high in this kind of cases. Like, this is one of the kind of its instrument. So now Professor will give you the brief. Back to you, Professor.
All right, I think thank you, Arya, for giving overview of this technology. Again, a lot of experiments are happening. We are trying to utilize the potential of this technology for deep proteomics and the quantitative proteomic analysis. Let's move on in the last section about data analysis part, because data analysis is very critical at the end. And many of the students or the participants of MOOC, if they want to analyze the data of mass spec at their home or using their laptops, how they can do that. So there are some new open access software coming up, which is like the FragPipe or MS Fragger, which is quite easy to do that in your PCs. So the next session is going to be talking about data analysis, and where our teams are going to show you how to take the your DD or DIA files from the mass spec and use this open access software, which is the MS Fragger. I will uh, just show you, like, how the software logo and things like that, so that if you want to make yourself familiar, you can do that also. But idea, this software is done from one of the academic lab, and here the things are so fast that you don't need too much RAM from the clusters, and you can directly use with the limited RAM and the computing power of your laptop, and directly analyze the any kind of workflow of DDA and DIA. So Amit, please share your screen and just briefly show the frag pipe data analysis.
Okay, thank you, sir. Yeah. So, yeah. So I guess you can see my screen, and here you can see the interface of the FragPipe software, the FragPipe version 23. Now, let me just give you the very minute brief of what is the interface looks like. So this is the basic interface. And one of the most important thing, whenever you are going to analyze any kind of raw files in the FragPipe, you need to set the path very. You need to set the path. So here you can see I have kept two different raw files here. That is came up from a rat, that is based on some rat tissue samples. Now, in case of this kind of analysis, we need whatever the path you are using in this entire path, in any name there should not be any space. So you can see there is a name NPTEL 2025. There is no space. There is an underscore, and even the output files you don't need to keep any space here. If you keep space, the software will stop analyzing the samples. Now, the very first thing is that you need to first upload the data files here. So here is the workflow tab. So yeah, basically here is the workflow tab. We'll add files. Now you will go to your folder. So basically these are two raw files you need to upload. You select this, and you can see these files are uploaded. These are DIA datasets. You need to put set DIA. Now you need to set the experiments here. So and also if these are bioreplicates, you can also set what are the replicates. It's a control sample or a disease sample. You can also set it. For now, I am setting this for consecutive. Now you need to also upload the different workflow. So it's a DIA file. You need to upload the DIA spec quant. Okay. Now you need to keep certain RAM and at a certain parallelism. So if you want to try out, please see what is the core of your system and set the parallelism based on that. Now then the next thing is the database. So we are using a RAD database. So basically the entire database you can upload with the help of UniProt. Now you can just browse it. I have already downloaded the database here, the RAD database. I'm going to upload this database and select it. You can see this is uploaded, and you will add decoy. So decoy is basically an completely opposite of the entire FASTA file of the RAD proteome. So basically you have to find out any kind of false to remove any kind of FDRs there. So this is basically a non-existent database that will basically try to match with your original databases and remove the false positive sequences of the proteins. It's taking some time to upload database. So after the database is uploaded, you can go to So you can go to just MS Fragger and you can set or what are the different proteins that you are like. I am selecting a trypsin. You can also select lyc or anything. You can also change the miscleavage. Then we can go to validation. So in validation, this peptide spectrum validation is important to see any kind of PSM is there. And then you go to run and you set the part that where you actually put the output files there. Yeah. So we'll just keep the directory set here and you will run. Yeah. So as you can see the file starts running.
All right. So great. Thank you, Amit. So what students can understand here that you've seen how quickly the entire process could be achieved, and this file will take maybe good half an hour trying to run, but earlier the same files used to take maybe several hours to day time to run, but now it can be done in just very short time, in less than an hour. So another software which will now show is Mascot matrix science, which Ankit will briefly walk you through and show you, like, if you have a very simple mixture of the protein, you want to identify their sequences and want to know what is the bigger hit, how to utilize that using the Mascot software analysis. So you'll do that, Ankit. Amit, you can unshare, yeah. Ankit.
Hello everyone. Yes. Yeah. So I'll show the interface for the mascot. So we had talked about a lot, right? Like getting the peptides and the mass spec. So now how we had identified, so PracPipe was one of the software. So in the matrix science mascot, so this is very user friendly and easy to use software for files that are not so much complex. So what we can do is that, so first of all, you can just register here with your email ID and give the search title. So here we are using two databases. So since if we are not aware that what is the particular sequence that contains from which organism, we can give all the Swiss prot and some contaminants which are general happens when while you are doing the kind of digestion and other processes like keratin and all can come from different sources. Then taxonomies, we can give all entries. Enzyme, you can add like trypsin. Then we are not adding any kind of quantitation or cross-linking as such, and then you can simply add suffix modification like IA and those for those things which we had already shown in peptide digestion. And then here we can go to do like oxidation, but if you want to have any ppms and all, you can add it. So then you can keep it all default with all the standards that are present, that are community based standards. Then you can choose your data file. So it is taking in the .mgf, that is mascot generic format. I have already uploaded it, and then you can simply start search. Okay, so it will start running and it will give us the result, and you can see so the y ions and b ions and those things that we talked upon, like getting the actual peptides. So here you will get that after adding that the identification of the peptides for a particular protein, you will see that for this particular protein, this number of peptides have been identified. And now if you go and click for any particular peptide, so you will get the fragmentation spectra or pattern as well, like the ones that I was telling
All
that
right.
for the
Thank you, Ankit.
peptide. Yeah.
So this is a very quick one, but at least students got a feel of it, how to do different software for analysis. You can stop sharing your screen. So just in the final minute or so, students have been also asking us in which way the proteomics is really, getting more and more transformative using the new mass spec technologies. So what I'd like to mention to you that our ability to really look at the proteome from the very small amount of protein, for example, as we just show you in the timsTOF, using 200 nanogram or 300 nanogram amount of peptide, one could now look at a complex proteome to more than 8,000 and 10,000 protein. Now we are actually started to reach towards understanding some key biological questions. Can we now do single cell proteomics from the single cell? Can we look at the cross links of the membrane protein and trying to look at more of the druggable targets? Can we look at even the imaging of the cell and use the spatial proteomics now as the new way of looking at where all the our major protein changes are happening? So there are many new workflows are emerging, and thanks to all these new instrumentation, new technologies of mass spec, which is now making the proteomics to a very, very unprecedented scale, which was not possible earlier. And now we can see different type of applications are going to be really quite important for the clinical as well as biological applications. I hope students who are taking the MOOC courses on proteomics and proteogenomics are keeping the pace, the new technologies that are happening, and you're reading them well. And if you have additional questions and queries, please do get in touch with us in our separate Telegram groups or in the chat, like different type of chat which we do on your forum. We are quite available, and do make this kind of forum as a more opportunity for your live interactions with us. I think some of you have also asked us whether you can get some opportunity for doing workshop in our lab. Of course you can, but you have to be the top performers in your course. And then we do give opportunity as an internship or some of the workshop we conduct pretty much free of cost for the student to come and learn those technology directly in our lab. But that we cannot give to everybody. That has based on the selection process to the top performer students. So wish you all the best for your exam for the Proteomics MOOC course students, and Proteogenomics students. We will have one more session with you in the next month. Thank you very much, everybody, and goodbye for now. Thanks the TAs for all the live session, very nicely interacted and well coordinated. Thank you, team.